Yeah, thank you so much for having me here to speak to you about E. coli. So for the next few minutes, I'll explain to you how we use E. coli genomics to track sharing across interfaces. We just call it a fancy name, really, doesn't matter. I come from ILRI. ILRI is a livestock NGO. We speak livestock for livestock. So what are interfaces? Interfaces are essentially points where systems or diverse groups interact. For example, we have an interface here. This is an interface. And this could be defined as zone of disturbance, zone of interaction, could be a zone of genetic exchange, pathogen transmission. And in complex environments where most of us come from, like in urban areas and rural areas, these are ideal for analyzing the interaction of pathogens at the human-livestock interface. However, there's very minimal evidence regarding the frequency, the pathways, and the magnitude of transmission of pathogens across these One Health interfaces. And E. coli is the perfect organism for that. It's heterogeneous, at the same time, a commensal. For example, in this one here, this is an example of an interface in Western Kenya where you see people living the livestock in the same household. Another interface in Southeast Asia, you see humans and animal source foods come into contact. So, again, tying up to AMR, all of us know AMR is the next big thing, and we should be very much worried about resistance of bacteria. And to study the transmission of pathogens at these interfaces, we chose Nairobi, which is the capital of Kenya, and Nairobi typifies any other LMIC city, be it Dhaka, be it Lusaka, whatever it is. It's a system where we have high disease burden, high antibiotic use due to rising incomes, ease of antibiotic access, and all that. And livestock keeping is very common. And again, this is, a very common scenario in most LMICs where you see the wealthy and the poor living alongside each other, very complex food systems, and livestock keeping is very common. That's one of our colleagues doing fieldwork. So Nairobi is the perfect place to do this kind of work. So the aim of our study was to investigate the population structure of E. coli in Nairobi to quantify the transmission of pathogens across those One Health interfaces. And our hypothesis was that basically everything is everywhere. That is, the distribution of bacterial strains and the genetic pools are limited to particular defined ecological niches, in this case, a household, as compared to an sort of cosmopolitan distribution of genes. So our household in this case is our study interface where humans, livestock, and peridomestic wildlife, they are all of them cohabiting sympatrically. So that's Nairobi. I live somewhere on the that's our national park. Is this working? Yeah, the white blob there is our national park. So again, feel free to come and see wildlife in the city. So we took the city of Nairobi, divided it into different socioeconomic status. The red, those are sort of poor slum-ish areas, all the way to the affluent areas, which are in green. And then for each particular area, we chose representative sublocations to represent each wealth category from highest to lowest. And then we chose 3 households. So we basically chose that 3 sublocations across the entire city, representing the wealth categories. And then we chose 3 households in each sublocation, one with large livestock, that is essentially cows and pigs, one with small livestock, that is chickens and goats and sheep, and one with no livestock to be our control to test the hypothesis that livestock is a risk to human health. And then we scaled that up across the 3 sublocations. So this is critical because it forms the basis of our work. And then for each sublocation, we collected biological data from healthy humans and healthy livestock and wildlife, food, environment, anything you can think of, really, we collected that. And then a lot of metadata, or should I say data, which links to socioecological dynamics, ecological practices, risk practices, and all that. And the work took 13 months. It's already published already, if you're looking to read more about the paper. So those are our samples. I said fecal samples from healthy humans, livestock, and everything else. of course, the normal pipeline of analysis, single isolate microbiology, sequencing, and what you'd expect for genomics. So in terms of our numbers, the majority were, of course, livestock. We had up to 13 different livestock species, from poultry, which includes chickens, geese, duck, and all that, goats, cows, pigs, rabbits, guinea pigs, and others. And then we had 421 wildlife samples, which was ranging from, I think, maybe more than 30 wild bird species to rats, a few monkeys, carnivores, not lions, I must say. So yeah, sorry about that. Just some typical mesocarnivores. So yeah, we had everything, and then 311 humans of all ages and gender. So we collapsed all those species into functional groups according to what we think E. coli would look like in these systems. So we have humans, livestock, bird, all the way down to wild mammals. Yeah. Okay. So now to our first finding, which is this fancy tree here, which again says almost that it doesn't matter. That is, all of the world is in Nairobi, and all of Nairobi is in the world. That is, the inner ring, which is the inner one, those are the sequence types. We found up to 537 sequence types out of 1,000 plus genomes, which signifies the level of diversity in our data. The middle ring represents the host source, and you can see there's no cluster, not like my previous presenter with the clusters. There's nothing here. And then the outer ring represents the Claremont phylogroups. Again, there's very minimal clustering across all that. So the first finding is that there's just so much diversity, and everything is within our city. And then secondly, E. coli is democratic. It doesn't matter whether you're poor or rich. You have similar types of E. coli. I think we should learn from E. coli about that. And then the second thing was to think about sharing. What do we mean by sharing? You can find sharing in many ways. One, we use core genome sharing, that is sharing of basically the actual genome itself. And then the resistome, which is defined as the collection of the AMR genes someone had. And for core genome sharing, we use CGMLST. I'll explain that a bit later. And then for resistome, we basically took the genes within an isolate, created a Jacquard index, and then if you had one, we defined your sharing. And one basically means that you have the same genes with another pair. And then, of course, when you say sharing, he has to... Oh, gosh. That wasn't me, sorry. Okay. And then for resolution, as you can imagine, it varies all the way with the level of resolution. That dude there, if you use MLST or PFGE, you say it's the same person. But the moment it increases resolution, they're actually two different people. So we used the highest level of resolution to define sharing. And then, again, back to the core genome sharing. As I said, we used CGMLST simply because CGMLST was a bit more robust and also, at the same time, conservative, allowing us to avoid bias due to alignment issues with a reference genome for a very diverse species. So we use CGMLST, and the first finding is that, so this histogram here, it just shows you on the x-axis, the CGMLST allele pairwise distance all the way from 0 to 2,000 plus, and then the y-axis is the count of the number of pairs. And you can see, and then it's split by whether you're from the same household. So all the pairs, we match them whether from the same household or from different households. And you can see there's a peak there under 10 for pairs within the same household as compared to different households. And so we used 10 to be our natural definition of sharing. I mean, you can argue that if E. coli acquires one single SNP a year, those are 10 years and all that, but we chose 10 based on that. And then, of course, we quantified to be sure that it was a sufficient number. And then in total, we found 150 pairs of isolates which were under 10. And that will form the basis of our discussion just now. So the first finding we saw was that If and when sharing occurs, it occurs within the household. So remember the household interface I mentioned initially? If sharing occurs, it's confined within the household sharing, which, if you think about COVID, it followed that exact pathway, that there was more sharing within confines, be it household, be it a school, as compared to between areas. So 3/4 of our isolates, our pairs, the 150 pairs, were all within the same households, and similarly within the same host. That is, if sharing occurs, it occurs within the same household and the same host, as compared to inter-household, inter-host sharing. And the second pairing, the second finding is that sharing did not matter with the E. coli lineage. You could hypothesize that it's only one lineage sharing, but actually it wasn't. So every dot here represents a sharing event, and basically says that sharing event was equally distributed across all the E. coli lineages. And then you can also hypothesize that for sharing between households is limited within the geographical distance. That is, household A and household B, if they're close to each other, they're more likely to share. But actually it wasn't the case. We found that inter-household clonal sharing was not associated with distance. As you can see, human-to-human sharing. For example, you can see that, which one is that? Yeah, you can see there's I'm not sure what color that is. For the second one, there's a very long dispersal event between human and LB, which is livestock, live birds, yeah. And you can see there's that long dispersal event, but generally there was no spatial signal. And then the more you sample, the more you see sharing events. Of course, that's a 101 of epidemiology, that numerators don't matter. What matters is the denominator. So for us, we had to go back to epidemiology 101, which is counting. And we generated all the expected, so the observed number of sharing, which was 150. And then we generated the expected sharing event. That is, each isolate in our database had an equal chance to be shared with another isolate. And then we generated our denominator based on the sharing event. And then you can see in the within household sharing one, sorry, in the first cartoon there, that's how we calculated our sharing events within the household. That is, if you have two isolates, that would be 2 times 1, let's say 4. That would be 4 times 3 over 2. Then between households would be N1 times N2. And then we had a list of all the possible pairs, but then we have a problem, which is a graph problem. That is, you have this mess here, in the first one. Everything is connected with everything else. And that's no, again, epidemiology. You have to find a way of reducing that bias of counting. So we use some algorithm which corrected for that. And you can see it there. So rather than having those 3, it changes to only 2 because they're the same. So you'll be double counting if you count all 3. So it's a game of counting, really. And what we found is that, and then we did bootstrapping to get the intervals and all that. What we found is that, so this is the expected sharing across the city, partitioned by within household or between households. And we found that if you look at the within household sharing, the only thing which sticks out, the ones with the red means more than expected sharing. The blue means less than expected sharing. We found that the more than expected sharing within a household was for some species in the clone, chickens and cows and all that. But look at this other side. There's very minimal, more than expected sharing events between households, which again confirms that sharing, if and when it occurs within the household confines. But we have a problem, AMR genes. AMR genes don't follow those patterns. I won't go into the details of all that, but the findings are for AMR genes, these are AMR genes, the JAK and index I mentioned. For the AMR genes, you can see the difference. There's no red here, but look at this. There's a lot more red on the between households. That is, we have more than expected sharing between households. And that makes sense, because in this city, we have people and humans are using almost a similar types of antibiotics. There's co-usage of antibiotics. There's co-sharing with the environment and possible plasmid involvement in all this. So this is something which we need to dig deeper to understand why are we seeing no pattern in the resistome. And then, what about the risk of keeping livestock? Remember our household structure, 2 keeping livestock, 1 not keeping livestock. We found 10 sharing events which were between humans and livestock. And again, you can see, for example, there's a chicken and a human there who are only separated by a single CGMLST allele. Basically, they're the same E. coli, which is interesting, but we only found 10. And there were almost all of them keeping livestock, apart from 2. And then the majority were also handling livestock. And they were all men. So that's a clear finding for epidemiology in this room. If you're a man, keep off livestock. So what we found is that there's risk of sharing, but not always due to livestock keeping or livestock contact. And then how about the AMR genes? So we look at the AMR genes epidemiology, and what we found is that we found up to around 70 different AMR genes which confer resistance to all kinds of classes. We can think of from fluoroquinolones to beta-lactams. Again, these are healthy humans, healthy livestock, healthy wildlife. And those genes were mostly found on clusters, building up quite fast to form these large networks, which we think is due to plasmids there. But then interestingly is that if you partition your gene similarity by livestock keeping only for humans, you'd hypothesize that people who keep livestock are exposed to more genes than those who don't keep livestock, or people keeping livestock are exposed to different kinds of genes than those who don't. But actually, that's not true. It didn't matter. If you look at this PCA diagram here, it doesn't matter whether you're keeping livestock or not. You add the similar types of genes. But if you are keeping manure, livestock manure, in your household, as a human, you're more likely to have more AMR genes. And therefore, we're thinking that the risk of acquiring genes from livestock is not because of keeping livestock. It's because of these other practices like manure management and all that. So in conclusion, we found evidence of recent clonal sharing between humans and livestock, and we show that E. coli population structure in this human-animal interface is primarily shaped by by household and household type. Again, typifying what diseases would do. And then secondly, similarities in the resistome of the isolates did not match the patterns of clonal sharing, but rather were more reflective of usage patterns or possibly plasmid transmission and all that. And then lastly, the impact of keeping livestock on AMR carriage, human AMR carriage, was mostly due to animal manure practices. And we support the hypothesis that everything is everywhere, that is, E. coli is across the entire city, but it's limited by the environment, which in this case is the household and the host. And therefore, confirming that hypothesis formulated back in the Netherlands in the 18th century, that everything is everywhere, but the environment selects. There's a lot of people involved in this work. I can't thank all of them. But I have to thank my co-founder author, Brian Wee from Edinburgh, who we worked a lot with. And then the funders, MRC in the UK, and Welcome for giving us the money to do all this amazing work. Thank you.